Hello and welcome students to the SRBPS Virtual Classroom Program. I am your biology educator Prerna Rathi. I hope you are all making complete use of our YouTube channel. Now let us come and see what I have got for you today. So here we are yet again to continue genetics. Let's start today's topic. We'll be beginning with linkage. Linkage children was discovered by T.H. Morgan, yes, Thomas Hunt Morgan, and he was working on Drosophila melanogaster, that is your fruit flies. What he did with children, he crossed a mutant type of Drosophila with a wild type, and in the F1 progeny he received the wild type because that was dominant. Then when he crossed it, self-crossed it, in the F2 progeny, instead of getting a standard dihybrid ratio of 9:3:3:1, he got a deviated ratio. उसने देखा कि भाई जो नए रिकॉम्बिनेंट्स बने थे ना, ये 3:3 वाले, ये तो बने ही नहीं, कुछ और ही बनकर आ गया. मतलब जो पेरेंटल था वही ज़्यादा आ रहा है, रिकॉम्बिनेंट बहुत कम बन रहे हैं. Therefore he came on the conclusion that these genes were linked. The gene for the body color and for the eye color were linked and hence were always inherited together. Such an inheritance is called linked inheritance. Such genes are called linked genes, and this phenomena, my dear children, is known as linkage. Can you see here in this particular diagram how the yellow and white were crossed with wild type, and when he received wild type in the F1 on self crossing, what did he get? 98.7% parental percentage and 1.3% recombinant, which should have been 62.8 and 37.2 respectively. The ratio should have been 9:3:3:1, where if we calculate parental are 10 and 6 are recombinant, the right hand side ratio should have been there, but it was not there. So this proved the phenomena of linkage. Okay, so these later on this concept of linkage was used by a student of T.H. Morgan called Stu Twent, who designed gene maps, DNA maps, linked genes की position से उसने इसको use किया to create gene maps, which is a very good thing. The next concept that we have to discuss today is mutation. What do you mean by mutation? Mutation कोई भी change है DNA में which is expressed in the form of a phenotype and is always inherited. The phenotypic variation that occurs due to change in gene or DNA sequence is called a mutation. The organism that undergoes mutation is known as a mutant. तो किसके अंदर होती है mutation? उसको क्या कहते हैं? Mutant. जो mutation cause करता है उसे कहते हैं mutagen. Agent of mutation is mutagen. Okay. What can be the mutation? It can be loss or gain of a segment of DNA. कुछ DNA का एक piece lose हो जाए या gain हो जाए. This can cause chromosomal changes, chromosomal aberrations, और gene mutation हो सकती है. एक point mutation हो सकती है, जैसे sickle cell anemia में GAG की जगह GTG हो गया. That is a change. Apart from that, there could be frame shift mutation where certain part of DNA could be added, or certain, even if one base is deleted, that would shift the frame of reading by one. The next phenomena that we have to discuss is pedigree analysis. Pedigree analysis क्या कहता है? ये सारी genetics की जो study है, it wouldn't be meaningful if we don't use it for the right purpose. So pedigree analysis is one such application of genetics where we find out or where we trace back the presence of a trait, the arising of a trait in our previous generations. अगर हमारी blue बहुत सुंदर सी आँखें हैं तो हम trace back कर सकते हैं कि ये नानी की family से आई हैं कि दादी की family से आई है. Similarly, we can also trace back certain disorders. As you can see on the right hand side, these are various symbols that are used while we draw the pedigree charts for various diseases. What are genetic disorders? Genetic disorders are disorders of the DNA or genes. They are categorized into two categories: the Mendelian disorders and the chromosomal disorders. The Mendelian disorders are those which follow the Mendelian principles of inheritance. Mendel के सारे laws, law of dominance, segregation, independent assortment, सभी को वो follow करते हैं. For example, hemophilia, color blindness, cystic fibrosis, sickle cell anemia, phenylketonuria, thalassemia. These are all the examples of Mendelian disorders. Then we have chromosomal disorders, which are caused due to the absence or excess of abnormal arrangement of one or more chromosome. या तो एक chromosome extra हो जाता है, या एक chromosome delete हो जाता है, या पूरा set of chromosome extra हो जाता है, पूरा set of chromosome delete हो जाता है. For example, Down syndrome, Klinefelter syndrome and Turner syndrome. Let us start with the inheritance of hemophilia. Hemophilia, children, is known as bleeder's disease. इसमें क्या होता है कि person having hemophilia will bleed to death from a simple cut. एक छोटा सा कट लगेगा क्योंकि उसकी बॉडी में कुछ clotting factors होते हैं जो ब्लड में fibrinogen, fibrin, thrombin, clotting factors नहीं present होते हैं, which have to start the cascade reaction of clotting. Those factors are absent. In the absence of those factors, a person will keep on bleeding. Can you see here? A male would be affected only if he has one X, because he has only one X. तो male तो XHY में affected हो जाएगा, लेकिन अगर female में XHX आएगा तो she would only be a carrier. So there are chances, it is normally transferred, from mother to son. If this woman bears a son, that the son, can you see here, 25% chances that, if a normal male marries a carrier female, then their children could be non-affected. If they have a son, their son can be affected and the daughter can be a carrier. Okay, children? Also, this is a very rare chance कि ऐसा भी हो, where affected male would marry a carrier female, then only the daughter would be affected. But this is a very rare chance that's what female becoming hemophilic is extremely rare. Okay, and affected transmits the disease only to the son, not to the daughter. That's जो affected होगी वो अपने son को disease transmit कर देगी. Daughters can receive the disease from both mother and father. The next is color blindness. Color blindness में क्या होता है? This is also an X-linked disorder. The inheritance is exactly similar to hemophilia. Okay, person suffering from color blindness is not able to distinguish between a few colors. जिस भी color के cone cells उसकी body में नहीं produce हो रहे होंगे, red, green, blue cones, these are the three basic colors which combine to form any color that you see outside. All the apprehensions, all the comprehensions about the colors that you make are made by the help of these three colors. So color blindness में these cone cells are absent and that person is not able to see that particular color. Next is sickle cell anemia, which is caused due to a point mutation जहाँ पर normal condition में RBCs की shape biconcave रहती है, लेकिन अगर body में oxygen stress या oxygen deficiency की condition आ जाती है, then as you can see the RBCs become sickle shaped and they coagulate the blood vessel. ये आपस में फंस जाते हैं, blood vessel को coagulate कर देता है, can cause bursting of blood vessels as well. This is the condition where glutamine GAG gets converted into valine because of GTG, and a normal hemoglobin A becomes hemoglobin S, which is affected hemoglobin. The next is Down syndrome. Very clearly if you can see Down syndrome में क्या है, सभी 46 अगर हम देखें तो सभी दो हैं except for this. Chromosome number 21 की 3 copies हैं. This is known as trisomy of 21. Chromosome number 21 की 3 copies आ जाती हैं, which causes this disorder where the mental retardation, physical retardation, open mouth, characteristic palm creases are there, furrowed tongue and tongue bulges out. All these are the basic traits that are shown by a person suffering from Down syndrome. Apart from that, other chromosomal disorders are like Klinefelter's and Turner's syndrome. Okay, so that's it. I hope everyone has understood. I hope you have all understood what I taught you and you are making complete use of this YouTube channel. Until next time, take care, study hard. Goodbye, children.